Okay, hello, cool people. So this is the first video of carbohydrate catabolism. So we'll have two pathways today, glycolysis and fermentation. After watching this video, you should be able to state the cellular locations for glycolysis and fermentation in a eukaryote. So we're going to focus on eukaryotes. These are organisms that have a true nucleus, for example, humans. I guess we should say we're focused on eukaryote heterotrophs. Again, humans. So anything that is humans, I don't know, whatever. You should be able to describe the chemical logic for the three irreversible steps in glycolysis, draw the substrates and products for the three irreversible steps in glycolysis, and state the number of carbons in each metabolite. And then just state what the enzymes are for each of them, and write the overall chemical equation for glycolysis. This is going to be pretty standard for all of the pathways that we talk about, where you'll need to know the chemical logic, the irreversible enzymes, the substrates, and the products that go with these irreversible steps. And again, irreversible means that in this case, there's going to be a negative delta G. They're not going to be able to go backwards, which is one of our main themes of metabolism, is that all of the pathways have to proceed in an exergonic direction, which means they can't individually, each cannot go backwards. And then we're going to talk a little bit about fermentation and its purpose, and you should be able to describe two types of fermentation. All right, so what is glycolysis? It's a catabolic pathway that converts glucose into pyruvate. So the overall reaction for glycolysis, I'm just going to draw out glucose because it's one of the molecules that you'll need. Down, down, up, down. I always draw alpha glucose, just so you know. Okay, and you should recognize here that glucose has 1, 2, 3, 4, 5, 6 carbons, and every carbon has a carbon-hydrogen bond, which again is another theme of metabolism, is that we break down organic carbon molecules, and obviously glucose is no exception. There are 1, 2, 3, 4, 5, 6, 7 CH bonds here. Okay, so in the process of breaking this down, because it has carbon-hydrogen bonds, those electrons have to be transferred somewhere because you're not on fire inside of your cell, and they get transferred to NAD+, which will make NADH. We're also going to have some substrate level phosphorylation, so that requires ADP and PI to be put in here. And then, so these are all of the overall, net molecules that are going in. And what's coming out is pyruvate, again, a molecule that you need to know how to draw. Okay, and notice that pyruvate here, this is pyruvate. Its abbreviation is PYR, all lowercase, and it has 3 carbons. We're going to get out NADH and ATP and water. And why am I forgetting my little plus signs? Did I forget anybody? Oh yeah, H+. Okay, so if you notice that glucose has 6 carbons, whoops, hey man. It has 6 carbons, and the carbon metabolite is going to go from glucose to pyruvate. So in order for us to balance out our number of carbons, we know that we're going to have to end up with 2 pyruvates, because 2 times 3 is 6. We have to get all of our we have to balance out our mass. We're going to make 2 NADHs and 2 ATPs, which means we need 2 of everything up here. Okay, and hopefully you'll have noticed that this is an oxidation reduction reaction. Why don't I just call it a redox? This is a redox pathway. All of our pathways are redox. And in our pathway, glucose is being oxidized to pyruvate. Again, we're on our way to complete oxidation of glucose, would be a CO2. So we're in an intermediate step. Pyruvate is an intermediate metabolite because it's not CO2, but it can help us get to CO2. So if glucose is being oxidized, something else must be being reduced. In this case, NAD+ is being reduced to NADH, which is our a reduced electron carrier, which will carry electrons eventually to the electron transport chain so that we can get, so eventually this NADH is going to go to an ETC or electron transport chain so that we can get ATP via oxidative phosphorylation. That was one of the ways that we can make ATP. But we also get, during glycolysis, we do get 2 ATPs via substrate level phosphorylation. I'm just going to put a P with a circle around it as a phosphate, so I'm just going to put 2 ATPs via substrate level phosphorylation. All right, so one of the things that you need to know is where glycolysis is located. Typically, we'll focus on eukaryotic cells, but I've included a bacteria, or I guess we don't really know if it's a bacteria or an archaea, so those would both be types of prokaryotes. And in bacteria and archaea, glycolysis happens in the cytoplasm, also known as the cytosol. Just in case you don't remember, this is the watery area that's all around all the other organelles. In a eukaryote, again, eukaryote means true nucleus, the glycolysis happens in the cytosol as well, or cytoplasm. These are the same words. I think I more often use the word cytosol than anything else. But yeah, so glycolysis happens in the cytosol of eukaryotes and in prokaryotes like bacteria and archaea. Where do you get glucose for glycolysis? There's 2 main places you can get glucose. One is your sugar storage in the liver, which is also known as glycogen. So glycogen can be mowed down. Remember how I can chop off the ends, the branched ends of glycogen, and mow them off to get to individual glucoses, and those glucose units can go on to glycolysis. Or you can just eat it. You can just, I don't know, have some cotton candy or something that has an apple. So you'll get it. You'll have the sugar being in your stomach, and then it'll move from your stomach into your bloodstream, and then it will go into a cell that needs it for glycolysis or to start glycolysis. Okay, so there's 2 main ways. You have some energy that is stored as sugar in your liver, primarily in your liver, as glycogen, which can be broken down into glucose units, which can then enter into glycolysis, or you eat it. Apple, I don't know, I think of anything that has a carbohydrate in it, that can be broken down into glucose and then eventually get into a cell for glycolysis. So glycolysis is a pathway, and it has 10 steps. Yikes, sorry about that. We're actually not going to go through every single step. We're only going to go through a few of them. So it has 10 steps. It has 10 enzymes. So when we do metabolism, we don't typically show the enzymes every single time, but wherever there's an arrow, there's an enzyme. There's an enzyme catalyzing that reaction, otherwise the reaction wouldn't proceed at any reasonable rate, and we all just would not be here. So just so you know, these are all enzyme catalyzed reactions that move functional groups or perform hydrolysis, ligate molecules together, do redox reactions. There's all sorts of enzymes floating around, and each enzyme is specific to its step. So there's 10 enzymes, and glycolysis literally means, so the glyco part means sugar, and lysis means to break. So all glycolysis does is it breaks the sugar, glucose sugar, in half. So if I have a 6-carbon sugar and it's broken in half, I'm going to get 2 3-carbon pieces at the end. Okay. We're also going to get, so what we're going to get out of this is we're going to get 2 pyruvates. These are our 3-carbon pieces. We're going to get 2 ATPs net, and we're going to get 2 NADHs. All right, so now we're going to draw. The slides that I posted for you are not exactly going to follow along here, so I just suggest that you get a piece of paper out and turn it the hot dog way. Why does that always confuse me? I don't know. Mine appears to be the hot dog way. And we're going to go ahead and draw out the pieces that you need to know. Okay. You're going to see this general drawing a lot, so get used to it. This is going to be the cytosolic membrane. So again, it's a lipid bilayer, which is why I have 2 lines, and I'm just going to kind of color it in a very beautiful way. The outside, this is going to be outside of the cell. I'm just going to call it the blood. You can call it extracellular fluid. I don't care what it is. The inside of the cell is going to be over here to the right. So this is going to be the cytosol. And we're going to assume this is a liver cell. And we're picking liver because it turns out your liver does a lot for you. That's all the pathways. And in your blood, there's going to be a lot of glucose, whether it got here because you ate 5 pounds of Twizzlers or because you're running low on sugar and you are starting to get sugar being released from glycogen. Doesn't matter where it's coming from. In order for glucose to get into the cell, you have to have a membrane transporter because glucose does not freely cross the cell itself. Did you know that? Hopefully you know that. So glucose here is going to enter the cell through a membrane protein, a glucose transporter. And I'm not sure exactly how I'm going to have to zoom in or zoom out. So now I'm going to start drawing the molecules that you need to be able to draw from memory. So the first one is glucose, and I always draw mine, again, in the alpha format. So I go down, down, up, down, H. There may be a point at which I stop drawing in all these OHs because they're not important. And the abbreviation for glucose is G. When you get to any of your assignments or your exam, you can always use the abbreviations where appropriate unless it asks you specifically to draw the structures. Okay, the very first step in glycolysis is irreversible. So it's a single-headed arrow. It's catalyzed by an enzyme called hexokinase. And I'm just going to abbreviate that HK. Hexokinase is the first enzyme that you need to know. And this is irreversible because it uses ATP hydrolysis, which we know is exergonic. So notice I'm putting ATP to ADP. So I went from a triphosphate to a diphosphate, which means I'm missing a phosphate. Hopefully you can see that I'm missing a phosphate. And the reason I'm missing a phosphate is because it's going to go onto the product to create I'm not going to draw, no, should I draw the rest of the oxygens? Should I, shouldn't I? Should I, shouldn't I? How much time do we have? Okay. All right, so if you'll notice that one of the phosphates from ATP was transferred over to glucose, which makes this product glucose 6-phosphate, or otherwise we abbreviate that as G6P. It's G6P because you remember number 1 carbon is here, the anomeric carbon. That's 2, 3, 4, 5, and 6. So the 6th carbon got the phosphate. Okay, so now we're going to talk about the chemical logic of this step. Chemical logic. We call this the chemical logic. Why does this happen, and why does it happen in the way that it is happening? Okay, so what color do I want? So this first step happens for a number of reasons. The first reason is that it traps glucose in the cell because that negatively charged phosphate cannot exit. There's no transporter to get G6P back out of the cell. Okay, so it traps G6P in the cell. This step also keeps the concentration of glucose really low because it's irreversible. We're making more G6P than G. It keeps G6P low, meaning that, or in effect, more glucose continues to enter. Okay, so if I have this guy at a very low concentration, that means more glucose will enter because it's going down its concentration gradient, and that is favorable. Okay. What else do I want to say? The first step in many pathways is irreversible, so this follows that. It's a point of major regulation, but this also serves to commit glucose to being inside the cell. Okay, is there anything else that I want to say about this? So this is the first irreversible reaction, which means its delta G is negative, and the reason it's negative is because we have ADP or ATP hydrolysis. What this does is it keeps the concentration of glucose low, so more glucose keeps coming into the cell, and glucose 6-phosphate is trapped inside the cell and cannot leave. Okay, the next thing that happens is there's an isomerization that is reversible, so there's a double-headed arrow here, where glucose 6-phosphate is isomerized to fructose 6-phosphate. And here's where I'm going to stop drawing the O's. So I'm going to put a circle around my P to indicate that that's a phosphate. I also have some O's here. And this is F6P because the phosphate is on the 6th carbon. And the enzyme that catalyzes this reaction is phosphohexose isomerase, or PHI, that stands for phosphohexose isomerase. So they're isomers because they have the same chemical formula, same number of carbons, hydrogens, oxygens, so on and so forth, but they're arranged in a different pattern. And this is not an irreversible step because it's a double-headed arrow, but one thing I am going to add to the chemical logic here is that the concentration of G6P being higher pushes, via Le Chatelier's, the phosphohexose isomerase reaction towards F6P via, I'm going to put LC, via Le Chatelier's. So because the G6P concentration is high, the equilibrium is pushed towards the product. So that is another piece of the chemical logic for having the first step being irreversible. If a subsequent step is reversible, it will push that reaction in a forward direction. So remember, all of our pathways need to go in a singular direction and whatever we quote-unquote called forward. You don't want to be going backwards with these pathways. Okay, the second irreversible step is catalyzed by PFK. Please don't forget this guy. It's very important. Phosphofructokinase. Yikes, that's a mouthful. Phosphofructokinase is also irreversible, and the reason that this one again is irreversible is because we're hydrolyzing ATP to ADP. And what we get is 2 phosphates now. And this is called fructose 1,6 bis phosphate. And if you remember, the little b, bis, BIS, is also a prefix meaning 2. So in this case, we have a fructose with 2 phosphates. One of them is on carbon number 1, and the other one is on carbon number 2. And what I kind of forgot to say about phosphohexose isomerase is that the reason that there's an isomerization right here is because it is going to set up for a symmetrical cleavage or hydrolysis because glycolysis is going to cleave the sugar or break the sugar into two pieces. So this one is getting us to a more symmetrical molecule for as symmetrical of breakage as possible. And again, you can kind of see it's not exactly symmetrical because the OHs are going in opposite directions. This one's down, down. I believe this one is up, and I believe that one is down. So we're not quite symmetrical, but it's getting more symmetrical. Okay, so the chemical logic for phosphofructokinase. 1, this is the rate limiting step. This is the slowest step. Which means it's the most, we'll call it tightly regulated. Okay. The concentration of F6P is kept low, so it pulls the reaction catalyzed by PHI forward. And the concentration of fructose 1,6-bisphosphate, or F16BP, is kept high, so it pushes the subsequent reversibles forward. And this is also, because it's the rate limiting step, this is the step that commits the molecules to glycolysis. Whereas the first irreversible step committed glucose to being in the cell, F6P to F16BP commits the molecules to undergoing glycolysis. Okay, so from here, there's that breakage or the cleavage of the molecule in half by an aldolase. And what we get are 2 3-carbon molecules. One of them is DHAP, and the other one is G3P. I'm not so... you don't need to memorize these particular ones, but basically we get 3 of them, or I'm sorry, we get 2 of them, and they each have 3 carbons. Okay, and then there's also an isomerization between the 2 of them, but it's the G3P that is carried forward. So I'm writing a really forwards and backwards reversible arrows, because there's a number of steps, so we're just going to skip right on past. And what you need to know is that all of them between G3P and the next one, which is called PEP, are all reversible. Okay, so this 3-carbon molecule is going to move forward. There are a ton of steps, so we're just kind of skip right over, but a few things happen in these steps. One of them is that we make an ADP, or an ATP. So in one of these steps, we're going to make an ATP. We're also going to make NADH from NAD+. Okay, so again, you need to know the 3 irreversibles, and obviously there's one reversible in between the first 2, so you're going to need to know that one. But between all these other steps that you don't need to know, what you should remember is that there's an ATP coming out and an NADH. And that leads us right into the last step, which is PEP. It's phosphoenolpyruvate. And that one is another molecule that you need to know the structure of. This is a high energy molecule, that's a CH2. Okay, so this is the last irreversible, and it's catalyzed by pyruvate kinase. And we get PEP out of it. Sorry, there's a negative right there. We get, I'm sorry, did I say we get PEP out of it? We get pyruvate out of it. PYR, also known as pyruvate. Again, 3-carbon molecule. And here is where we get an ATP from ADP. The third phosphate is coming from this one right here. If you'll notice what we had going into the symmetrical, after the symmetrical cleavage, were 2 phosphates. One of them will end up here, and the other one will end up there. Cool, huh? Okay, so what is the chemical logic of the pyruvate kinase step? This is the third irreversible. It's the last step in the pathway. Okay, and it effectively keeps the concentration of PEP low, so it pulls, via Le Chatelier, all preceding reactions forward. Okay, so before we move on, let's take inventory of what we have. So we've got glucose, which has 6 carbons. Okay, it gets phosphorylated by an ATP to make glucose 6-phosphate, which also has 6 carbons. And then there's an isomerization to F6P, which also has 6 carbons. And then there's a second phosphorylation to F16BP, which also has 6 carbons. And then there's the breakage step into 2 pieces, where dihydroxyacetone phosphate has 3 carbons. And then glyceraldehyde 3-phosphate also has 3 carbons, but it's the G3P that continues forward, and we have 2 of those. Okay, so we'll have 2 of those moving forward, which means I'll have 2 of these, 2 of these, 2 of those, 2 of those, and 2 of those. So our net equation is we had glucose going in. Where do I want to write this? Oops. Whoa, I don't know what just happened. Yikes. Pull that back out. Hey, why do you do that? Scrunch. Okay, so we have glucose going in and pyruvate coming out. I put in, it costs me, so I'm going to do a minus, it cost me 2 ATPs to get this whole thing started. The first one with hexokinase, the second one with phosphofructokinase. I got 2 ATPs back out in those intermediate steps, and then I also got 2 ATPs from the pyruvate kinase step. So overall, I have 2 ATPs. I also got 2 ATPs out of NADH, so I have 2 of those. And then I got 2 pyruvates out. Okay, so I can write again my overall equation. I had 1 glucose. I got 2 pyruvates out. I got 2 ATPs. I got 2 NADHs. And then in some of the mechanisms, we got some water out, and the H plus. If I created 2 NADHs, I would have had to put 2 NAD pluses to go in. To get my 2 ATPs, I have to have 2 ADPs and 2 phosphates also going in. Cool, huh? Okay, so those are all of the steps that you need to have memorized along with their structures and the chemical logic. It's a lot. There's a lot of information in glycolysis and in all of the other pathways. I don't think that any of it is necessarily terribly challenging. It's just a lot. So try and keep it organized. Okay, so proceeding on from pyruvate, we're not going to go too crazy far here. Actually, no, before I talk about pyruvate, I have 2 other things that I want to mention. Hey, how do I scrunch this? Scrunch, scrunch, scrunch. Okay, so throughout glycolysis, just so you notice that there was no O2 going in or out. Okay, so we would call this an anaerobic pathway. Okay, we also did not generate any CO2. So what we know is that glycolysis, we didn't generate any CO2, so glycolysis is not the completion of catabolism for a glucose molecule. If we put 6 carbons of glucose in, we're going to need 6 CO2s coming out somewhere, and we didn't generate a single CO2. So that pyruvate has to go on and do something else so that we can eventually get our CO2s back out. So let's talk about pyruvate. It's an intermediate metabolite, and it has a whole bunch of functions, and people typically talk about the 3 fates of pyruvate. So one of the fates is that it goes on to the TCA cycle or the Krebs cycle. Another one is that if it gets a nitrogen on it, it becomes alanine. Do you see alanine in it? NCCH. Okay, so we can get an amino acid out of it. And then the third fate is fermentation. Okay, so this one specifically is going to be lactic acid fermentation. And this is reversible, so it can go forwards and backwards. Okay, so lactic acid fermentation is not part of glycolysis. It's just the next step forwards. It isn't the Krebs cycle, and it's not making an alanine. Or specifically, maybe we'll just say, no, I can, yeah, what fermentation is. It's the completion of metabolism without using an electron transport chain or a terminal electron acceptor. And this is abbreviated TEA, like T. And we'll talk about electron transport chains and terminal electron acceptors many videos from now. But fermentation is a completion of catabolism without an electron transport chain or an electron acceptor. Fermentation is also aerobic. Anaerobic. So it does not use no O2. No use of O2 here. But what its purpose is to regenerate NAD+ for glycolysis. Okay, so if you'll notice, when I go from pyruvate to lactic acid, pyruvate is being reduced to lactic acid, or there's a gain of a hydrogen here, and that hydrogen comes from NADH, which is oxidized to NAD+. Okay, now I'm going to zoom back out and connect some things, and this is going to be really messy, but this NAD+ can re-enter or enter back into glycolysis and keep feeding glycolysis with a major molecule that it needs or major coenzyme that it needs in order to keep functioning. So fermentation, the definition is completion of catabolism. It's anaerobic, so it doesn't use O2, and the purpose is to regenerate NAD+ for glycolysis. So eukaryotes, humans, we do lactic acid fermentation. There is another flavor of fermentation, and this is the last thing, and that's ethanol fermentation. And this is done, yeast do this, which are also eukaryotes, but they're single-celled, and some bacteria do ethanol fermentation, where, again, there's glucose to pyruvate, whole bunch of steps, 10 steps, lots of steps, glycolysis happens. And then in ethanol fermentation, there's an intermediate step to acetaldehyde. and this releases a couple CO2s. And how you know is because acetaldehyde has 2 carbons, pyruvate has 3 carbons, because you'll memorize that. And the only way to balance this out is to have a 1 carbon unit leave. So 2 plus 1 is 3. So our 1 carbon unit that will leave will be CO2. And then finally, from acetaldehyde, H3, we get to ethanol. Just so you know, some eukaryotes, single-celled yeast, and some bacteria can do ethanol fermentation, and this will also, did I put that in the right spot? Nope. Hey man, go back. This will also obviously regenerates NAD+ for glycolysis. So just out of curiosity, are you thankful? Like, what would happen if instead of lactic acid, so this is what makes your muscles sore, the lactic acid or lactate, that happens during anaerobic metabolism. What would happen if instead, while you were like running or working out or doing something that requires sugar to be broken down, instead of making lactic acid and making your muscles sore, you would just get drunk? Woof. And then eventually your brain would shut down and all these other horrible things would happen. But maybe we should be grateful that our body doesn't ferment ethanol, and instead it ferments lactic acid. Okay, food. All right, so what this figure shows right here is that there are 3 irreversible steps, and this follows, again, one of the themes of metabolism is that a pathway has to proceed in an exergonic fashion, and I just happen to like this figure because it shows these big drops in energy for the 3 irreversible steps: hexokinase, phosphofructokinase, and pyruvate kinase, and everything else in between is about the same energy, and the only way to get it to all move in one direction is, again, keep this concentration low, so it pulls them all this way, and we keep this concentration high, so again, it pushes them all this way, and we proceed in a singular direction. Okay, so why are there so many dang steps in glycolysis? Biochemistry professors want to make you miserable? Oh man, I hope not. If there was only one enzyme in glycolysis, biochemistry would be too easy and anyone could get into med school. That might be true, but it's not. Lots of steps means that there will be lots of intermediates, which could be useful. Ding, 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 that one's true. And many steps means that there are many opportunities for regulation at the individual enzyme catalyzed steps. That is also true. So a lot of the intermediates can come and leave. So if you think about it, glucose to glucose 6-phosphate to F6P to F16BP, so on and so forth. Let's say that instead of taking in glucose, let's say you ate a lot of fructose, like high fructose corn syrup, then that fructose would be phosphorylated and come in sort of sideways at the fructose 6-phosphate section. And again, maybe that fructose 6-phosphate is needed for something else and it can be leaved. A lot of these intermediate molecules can be either siphoned off to go to other pathways, or something else can be entered in sideways and you don't have to start with glucose. Okay, that's all I got to say about glycolysis and fermentation. Have fun drawing, and I'll see you later. Bye-bye.